We got 10 people so far. Still can't pull. Got a fair number of people here. I'll probably get started. Okay, so this is in presentation mode, right? Yes. Hey, good morning, everyone. I don't know if we have anyone from Europe, but good afternoon if we do. So in terms of the agenda, uh, I'll go through essentially a summary of what's gone on since news on funding. And then we'll really get into the bulk of the meeting, which is really chemistry and modeling has really been the focus of this project in the early stages. And then end off with some updates from the variational and enamine collaboration. So they have some modeling results in progress, and really finish off with next steps on synthesis and testing of compounds. So you'll kind of recall from the last meeting, we were having some problems getting a batch of building blocks in through Canadian Customs. I'm happy to say we were able to resolve this eventually, and those building blocks were able to get into the hands of the chemists, and they've been working diligently to make those target molecules. And since the last update, the UHN team has been able to make just over 20 compounds, and those are awaiting testing at the moment. As well, in the background, they've been exploring some potential routes to make that core of the lead series asymmetrically. Currently, we're making it as a racemate, which I think is fine for this stage of the project, with the idea that any interesting compounds will separate out the more active enantiomer. But as this program progresses, and we hope that this lead series does progress to more interesting compounds down the line, we're going to need a synthetic route to make that active enantiomer selectively. So this is something that the team is working on in the background. We have some new modeling results. So we asked Conrad to kind of look at the B ring of that core and look for potential aromatic substitutions in that region. And I think this really led to some interesting results. So Conrad was able to identify close to 300 potential analogs that have good binding scores in that region, but also have the potential to pick up some additional hydrogen bonding interactions. So I think this is, if this is real, I think this is a really cool area of the molecule to explore. So for the backup series, Variational submitted their first two batches from their active learning exercise. One is from the build on demand space from the enamine library, and the second is looking at some additional benzodiazepine analogs. And some of the results that came out of that are actually commercially available compounds or known drugs. So we met with variational enamine independently to discuss next steps, and enamine has already started work on making those molecules from the variational exercise. So I did allude to the fact earlier that we do have a funding update, and I didn't want to really steal Rachel's thunder, but you know, congratulations to her because we were awarded a Kennedy's Disease Association grant. This is a 2-year grant, really focused on the modeling and the chemistry part of this program. Um, but a really significant milestone for the team that we now have some additional funding to help drive the project forward. So I know that Rachel isn't here today, but she will kind of reach out to the team when she has some more time to give some more details on this grant. But she's also going to be going to a conference put on by this association in the spring. So I think again, this is a really cool milestone for the team. So now I can turn it over to Frank to give an update on the chemistry. And I assume, Frank, I can just drive from here.
Yeah. Yeah, this is the first batch of compounds that we submitted. So I'm re-upping this just to show some preliminary ADME stuff that or data that we were able to acquire. So I just threw in a few compounds in there from a separate batch of compounds that we were submitting for ADME studies. So I just decided to submit 4 compounds here that had kind of the best PR56 activity. As you can see, solubility all less than 1 micromolar, and microsomes. So mouse liver microsomes had clearances of about 3,000. So that's relatively high. So it just shows it's being cleared in the liver quite rapidly. So these are some things that to keep an eye on and to optimize in the meantime as well as we continue to make compounds for synthesis. I've also included here BBB scores, so anything above 4 would be considered good. So we have some in this batch of compounds that were pretty decent, so that's good to see. Next slide, please.
So this mouse liver microsome assay that you did, I guess you didn't look. Did you do any mass spec on that to see what, these things are breaking down into, or it was just really looking at clearance of the parent?
Yeah, we look at clearance. So we're outsourcing this. We're
Okay.
trying to get our internal capabilities up and running. It should be go up and running soon, but in the meantime, we've been outsourcing this. And so they just look at kind of the mass of the analog and not so much the breakdown.
Right.
Okay. And as Pete alluded to earlier, our team here, chemists, have been able to synthesize about 22 compounds shown here. So we just dropped those off yesterday with Feng Ling. So it'll be good to see some results hopefully soon. As you can see, a lot of different analogs, both heterocyclic, alkyl, aromatic. So it would be interesting to see how all these behave and how these match up to kind of the computation results that were performed. And that's it from our update. I'll quickly just point out that yes, we are in the background trying to optimize synthetic routes. I won't bore everyone here with kind of chemistry details, but that's still ongoing. No fruitful results just yet, but yeah, we're working hard to get some of the asymmetric stuff going and maybe alternative routes to that benzodiazepine core as well.
Okay, so again, as Frank mentioned, these are now in the hands of SGC. So when Feng Ling has the opportunity, she'll test these compounds. Does anyone have any questions or comments on the chemistry update? Okay, so maybe I'll just move on to the modeling exercise. So, Conrad has, you have a comment, Conrad, or?
Yeah, sorry, I'm just from the doctor's office and I had a very multiple pain. I just had a question. Okay. I just wanted to know, Frank, what you use for insolubility? Because we're still having issues at SBC with solubility, predicting solubilities. I would like to have your values so that I can run some predictions and compare different softwares and see if I get something that it's a bit close to reality.
Sorry, what did you what? I couldn't.
I don't know. What method do you use for to get the solubility of your compounds? Because I saw you said some of them were below 1 micromolar. I would like to get those values and the respective compounds where the exact solubility are known, so that I can run some predictions and compare solubility from different programs to see which one we can proceed with from assigned.
Sure, yeah. I mean, I can send you the procedure that, again, the kinetic solubility calculations were outsourced. I can send you the procedural details if you like. Anything that's below 1 micromolar, I don't think you get, or at least they didn't provide like a specific uh, um, micromolar number, just because it's all less than 1 micromolar. But I can provide that for you if you like as
Okay. So yeah, I was going to give an update on the modeling as Conrad is be able to present today. He's out somewhere, but um, essentially what we asked him to do was focus on this aromatic ring um, to see what additional analogs we could make in this region. And what we asked him to do was just look at aryl rings right now, 5 and 6 member rings that carry the halogen. The reason we asked for one halogen, or potentially look at the commercially available Grignard reagents, is so that we could essentially follow along with the same synthetic strategy that we're using right now to make that diazepinone core. So what came out of the exercise that Conrad did was he was able to find close to 300 compounds that bound well in his pocket. But not only did they bind well, it's compounds like this that just have an aryl ring. Basically, that activity is just driven by phobic interactions. He found a lot of compounds that potentially can hydrogen bond in this region. So we have a lot of compounds that have potential to bind to the backbone, which I think really, if this is real, gives us the opportunity to really drive some potency down on the series. I don't want to go through obviously all 300, but I just took a few representative examples to show kind of what came out of that modeling exercise. So compounds like this, small molecule modifications here that carry a carbonyl group have the potential to form a hydrogen bond to this backbone glycine. So here we see this potential hydrogen bond to the amide nitrogen. Some other compounds actually have potential hydrogen bond donor acceptin interactions with the molecule and that backbone glycine. So forming not only a hydrogen bond to the amide, but potentially again to the carbonyl of this backbone glycine. Um, Thirdly, there's some compounds like this triazole that just have this one hydrogen bond donor that can potentially form a hydrogen bond to this backbone, or compounds like these aerosols that again have hydrogen bond donor acceptor interaction with this key residue in this binding pocket. So like I said, I think this is pretty exciting to explore this theory. Now, the one, I guess, caveat or drawback from having these compounds that have potentially an acidic hydrogen in that building block is it wouldn't be amenable easily to make a Grignard reagent when you have a hydrogen in the molecule. But there are ways around that. So I think sort of a protection approach should be feasible to make compounds like this. So really, I think for next steps, I've already gone through kind of a visual inspection and again, tossed out some molecules that may not be interesting and picked a few that I thought were interesting. But I think we really, as a group, at least with the chemists, need to narrow it down to probably 5 or 10 potential analogs that would answer this question of whether or not this is real, and decide which compounds to make. Or, you know, is there a different way to install this group? But it's certainly something I think that is worth exploring, and certainly can open it up to thoughts or ideas from anyone on the meeting today and what their thoughts are. Other ideas that we could focus on, blood-brain barrier scores if we wanted to pick that as a cutoff is another thing that we could look at. Or do we just go for the low-hanging fruit and kind of make some of the easiest molecules, like something like this? If we were to protect that nitrogen, it may be commercially available. If not, we could certainly just do that and make a molecule like this.
Pete, I was just going to say for discussion purposes that I like the idea of putting some polar groups in this because the leads are highly lipophilic and that could be contributing to, the quick turnover that we saw in terms of the metabolic stability. There's a general correlation between high C log Ps and just turnover in terms of metabolic turnover, in addition to picking up additional hydrogen bond acceptors within the pocket. I mean, my computational chemists are telling me that it's very lipophilic. So if you can make some hydrogen bonds to the backbone, that could be very helpful.
Yeah, no, I agree. Okay, so I think we'll just work internally to kind of, like I said, narrow down that list to some of the more promising and synthetically feasible analogs, and provided we have resources to make those molecules, I think it's worth getting started on trying to make a few of these. So that's kind of the update that I have from chemistry on our side. Guess I can stop sharing, and Pete and Brian, you can give an update on the variational work that's been done to date.
Yeah, and actually Dave from Enamine and I put together a joint presentation in terms of what we've done at Variational in terms of the design and what Enamine has done in terms of the actual synthesis. And so we've talked about this, so we'll kind of go back and forth. We just sent the slides to you probably a couple minutes before the meeting, so you feel free to post those afterwards.
Yeah, thank you.
Let me see if I can get the slides. Is that coming through? Okay. So Dave and I put together these slides. We also came up with a header page, which we can change as we go forward. We're just trying to align all of our efforts here. This is a reminder of the approach that we take to what we call a low data target like PRMT6, the allosteric site. There's really when we started on this, it was really the one, maybe two lead series, if you want to call it that. Basically the diazepinone series and some that are non-cyclized. And it was only about 46 compounds with a couple of crystal structures. And so the way that we approach this is different than your traditional DMTA cycle. We really look to do an enhanced DMTA cycle that's all computationally driven and driven through machine learning. Our machine learning algorithm is called Anki. Can generate lots of different compounds, which we then ground in a computational model, which we built off of the crystal structures to analyze those. And then we've done this in a couple of different ways. We wanted to constrain to the enamine space as a very practical area to try to get some initial compounds to begin to explore different scaffolds. And that's where enamine comes in, and Dave will talk about what they've done on that. And then we've also recognized that the diazepinone is related to a really old class of drugs, the benzodiazepines, for example. And so to try to increase the diversity of compounds with things that are, readily accessible from a synthesis point of view, we actually went and looked at, various benzodiazepine-like analogs and used those in the docking exercise as well. And so from those two different approaches, we've come up with ideas in terms of compounds to potentially look at for testing. Eventually, once we get enough of a data set, we can actually start thinking about doing a pure generative design where it's really trying to increase the novelty in terms of new chemical space. But we like to do this stepwise. What I show here is just and Pete will share these, so I'm going to go through this kind of quickly. All of the compounds that we put together for synthesis consideration. You can see that it does have the 3 aromatic rings, but it's gotten rid of the diazepinone, for example, and whether it has an amide here or it has the free amine. And again, this was constrained to the enamine space, so this is in their make-on-demand space, and so they have the scaffolds available for rapid synthesis to try to make this quick in terms of trying to find other scaffolds that may have activity. So this just gives a list of the various compounds. And I just made a copy from Data Warrior just to show the different compounds, the scores in terms of how they worked in terms of the docking as well. So that's what's shown here. Next, I just give a couple of docked poses. And if desired, we actually could send all the PDBs. One of our computational chemists, Alex, which you might have met before, actually has all those. So if you want them, we could also post those, Pete, to the drive or something like that. They tend to be large, so it'd be probably better not to send them through email. You can see this is the original lead, one of the ones that was crystallized. So that one's in purple. It's actually not showing up purple in this one, which is weird. Anyways, it was purple. Sometimes when you do this cut and paste, it changes things. Our compound, when I say our compound, the novel compound is in cyan. You can get a sense of the overlap, some of the hydrogen bonds, either water mediated or to the various different amino acids. So this is the overlay, and then this is the compound on its own. Here, this one's in purple. Okay, so here's again one of the standard compounds that was known. That's in purple, and that was the same as on the one before, but for some reason the color changed. And again, another idea that is in cyan that's overlap with those. I just picked a couple of representative docked poses just to give an idea of the process and a little bit of what it looks like. From here, I'll turn it over to Dave to talk through the actual synthesis.
Yeah, thank you, Peter. For everybody who doesn't know me, I'm Dave Hathaway, Director of Business Development for Enamine. And initially just selected 40 compounds from their modeling and provided it to enamine. For 7 of those compounds, enamine removed that did not appear to have feasible synthetic routes or required expensive intermediates. So we proceeded with 33 compounds. As Peter elaborated to, they restricted N-key to enamine's real space, chemical space designed using enamine's 300,000 in-stock building blocks, and we have about 170 validated parallel synthesis protocols that we've developed over the last 30 years. That paired with our in-house knowledge, it allows us to efficiently and affordably provide that chemical space. And overall, the synthesis is progressing as expected, maybe a little bit more efficiently than we all had planned, actually. We hope to beat the initial timelines, but even with a few expected synthesis failures, we're still on track to successfully complete about 70-75% of the originally targeted 33 compounds. Here you can see 16 compounds have already been obtained or are fully ready. 8 compounds are currently in different steps of purification. These should be able to convert to being available within the next 1 to 2 weeks. There's 1 compound that's still in synthesis, and 7 compounds that did fail synthesis or are on hold. At this stage, as we had previously discussed with Peter Sampson and variational, that we're taking more of a pragmatic approach. So instead of chasing down every failed synthesis, we'll focus on completing the compounds that are progressing and get them to Aquara for biological data. And Pete, can you switch to the next slide?
Yep, sounds good.
These are just the 7 compounds that had failed. And the 1 compound that we did put on hold. We didn't investigate whatsoever, but my hypothetical analysis just in the last 24 hours looking at them, it's just the installation of both heteroaryl arms on the central nitrogen failed. Either no reaction or very low conversion to the tertiary amine. So, of course, we haven't investigated it, but that's my initial thoughts. But that data, once we have the final compounds, will drive the next variational lead finding campaign or SARA iteration. So yeah, I think that's everything. I did have some logistics standpoints, Peter Sampson, in terms of if you did want partial shipments of compounds or if you wanted to wait until the completion of all successfully synthesized compounds, as well as logistically offline, we'll need to confirm where we will be shipping these compounds when they are completed.
Yeah, thanks, Dave. That's amazing progress. When I saw Pete's update, I didn't even realize you guys had started, so I think this is really amazing. I guess in terms of partial shipment or full shipment, what's sort of your timeline on kind of completing everything? And then for me, I think I've the Feng to see what her capacity is for testing the compounds. If she's able to run a really large plate, or how many compounds can you run at a time, Feng?
Yeah.
Now you've got about 20 compounds from you and potentially another 20 from enamine that I guess could be here in the next month or so.
It's okay. It's I can do that.
Okay,
yes.
One suggestion that you can consider, because these are more speculative. We don't know that any of these cores are active yet. You could consider doing maybe 2 or 3 concentrations instead of doing a full dose response. Maybe consider a couple concentrations, see if you see any activity If you do, then follow up with a dose response, because some of these may just be inactive. It's possible this whole series could be inactive.
Yeah.
So just offer that,
Hopefully
because
not.
we're really starting in new space. And so again, trying to make this as practical as possible, because we're really trying to explore different space. And then as we get activity, then we can hone in on that more carefully.
Yeah, very good point. Yeah, I will do that. Just the initial screening, just select the hits for the dose response test.
And if we're really lucky, they'll all be active. But yeah,
Yeah. We hope.
Mhm.
Yeah, so
Um,
Dave, I can send
Peter.
you the address for the SGC where these compounds will be sent. But again, what is your thoughts on timeline for this first batch?
So we have the 16 compounds already ready. Then the 9 compounds in purification or in synthesis. I would suspect that the 9 compounds will have completed, or we will see that they did not reach purification standards within the next 1 to 2 weeks.
Yeah, I mean, I think if you just ship everything at once in a couple of weeks I'm fine with that.
Yeah, great. And yeah, over the next week or 2, we'll keep you updated on the progress, and if anything delays, then we can adjust as necessary.
Yeah, unless it what's your capacity Pete at variation, or would you rather see these results like ASAP, or is a couple of weeks fine with you?
I could tell you that Handel, the CEO, is eager to see any results as quickly as possible, but that's, I understand the practical nature of it as well. I mean, he's actually thinking about doing fundraising. So any results he could immediately put into a slide. The nature of a startup.
Yeah, well, I mean, if that's the case, if you wanted to ship those like this week, then we could probably get those. I don't know, Fengling, what your capacity is, but if that
okay.
initial screen could be turned around quickly, then
Yeah, for me, it's okay.
And if it's not possible, that's fine, but I can tell you we will use the results if they come out positive as soon as possible.
Yeah, and just to mention too, right, we could ship them, I think, on Friday of this week at earliest, and it will take 1 to 2 weeks for delivery, right, coming from
okay.
Kiev.
Yeah, then let's do that. Let's ship the initial 16.
Okay. Yep. And Peter, I will just connect offline and we'll email you so that we get the correct address to ship it to. And Arena, my colleague, she'll be able to help us start that shipment.
Yeah, of course. Please, please send me a shipping address. So it will be helpful for me.
Yeah, okay. Yeah, those can be shipped directly to Canada. They won't be routed through the US, right?
Those will be routed through the US.
Hmm.
Yeah, unfortunately, yeah, I know. Yeah. Yeah,
Okay.
we don't need to talk about all that, but yeah, unfortunately, we have to import them into our facility in New Jersey.
Okay. Yeah, I think, well, if we go through SGC rather than through Agora, which they will anyway, then hopefully that expedites things and we won't go through the same issue we had when I tried to import them.
Yeah, and we'll label them in terms of just compounds for research and development only. So hopefully it will be pretty smooth
Okay.
customs wise with all the fun going on. So
Yeah. Well, I guess you'll need their customs broker, SGC customs broker.
yes,
But
correct.
we can talk about this offline.
Yeah.
And in terms of next steps, I mean, we'll wait for data to come in, either positive or negative. So as you get that, let us know. We will take that and put that back into the model so that we have experimental data, new experimental data, as well as any that's come from the original series. We'll import all that data again and then go through an additional cycle of design, bring those back to Enamine, and then we'll keep doing this iteratively over time. But we'll take a little bit of a break while we're waiting for the combos to be finished and for the data to be generated.
Absolutely. I think that's you know, like I said, it's really awesome progress. I know that Liz from Charles River is on the line. We just had a discussion earlier in the week, just thinking about potential to use their resources when they have some availability. So I think what I can do is we do have that diazepine batch that you had identified. Can send that to their team to see, you know, at least start thinking about if it's possible to make some of those, as well as the B ring synthesis. As if they and maybe Liz, you can chime in when you have some resources. At least if we start to think about um, that process, whether or not you have starting materials, whether or not it's feasible. I think at least getting ahead of that, so that when you do have resources, we could potentially pull the trigger on some of those targets.
I
Yeah,
actually
we
have
can
a
definitely take a look
slide
at this
with
and
those.
have a look. Sorry, go ahead.
I was just going to add, it'll be in here. I actually have a slide with the structures of some of those benzodiazepine-like structures.
Oh, okay.
Yeah, so maybe just jump to the next slide. Again, I just kind of lumped them all in here, but you can see, you know, some of the resemblances to various benzodiazepines, including known drugs, right? That maybe the issue with some of these known drugs is they're actually scheduled agents because they can be abused. But all of these compounds, I believe, are known, so there should be synthetic routes available to those. And again, the idea of this is to slowly move out the diversity from where we started, and offer you know, potentially additional starting points. I think that's all we had, though.
That's really cool. Thanks, Pete. Any questions for enamine or variational? Great. So I think for next steps, I don't really have a whole lot to say. We have a list of targets at UHN that they're still working through. Frank had sent me a list of building blocks. So I've asked kind of Rachel to see if you know what's available, so that we can order some of those building blocks to bring in. Those are still on that kind of C portion of the molecule that we've made most of the modifications at this point. And as Fengling said, she's going to be able to start testing some of those molecules. So hopefully we'll have some data, you know, later this month or early in December, that's going to help inform next steps on where to go. And hopefully we have some hits that we can sort of start moving further down the line, for additional testing and potentially some ADME or PK. But I think that's all I have for today. If there's any other comments or questions. But I will put together a summary of this meeting. I think for the next meeting, obviously we've got December coming up, so we'll meet again in January. But any pertinent information that comes out, particularly assay data, I will share that you know, once it's available. And you know, if we need to have ad hoc meetings in between to discuss something, then certainly let me know and we can arrange that. I guess one other thing that I should mention is we are having some law students do a preliminary freedom to operate on this series exercise. Some of you that have been on the M4K project, you know, you're kind of familiar with this group that they're going to go through on a SciFinder search to look what's available and whether or not there's any overlap with what we're doing in freedom to operate. So once they have done that exercise, I'll invite them to team meeting and they can go through those results. All right, if there are no comments, thanks everyone for joining, and again, congratulations to Rachel on the funding, and we'll see everyone probably in the new year, I guess.
Thank you very much, Peter.
Good to
All
see
right,
you all.
thanks everyone.
Yep.
Everyone, bye.